Thank you. I appreciate it, Marcella. Is that my cue to get started? Just checking.
Yes.
Awesome. Okay, great. Thank you so much for introducing me. That was really sweet. And thanks for everyone for showing up. I know it's kind of weird conditions today, but I'm excited to be presenting at the webinar. It's my first time presenting for AgBioData. And I actually heard about it at a conference I attended in July at a ASPB. So I'm really grateful that the stars sort of aligned to be here. And hopefully what I'll be talking about slots in with all of your interests in terms of, FAIR data principles and also data visualization. So the title of today's talk is Towards Sustainable Genomic Data Accessibility and Visualization with the Integrated Genome Browser. Like Marcella mentioned, my background is in forest and wildlife ecology. So I was doing population genetics works for threatened and endangered species. Essentially, I was trying to determine the current status of these species to inform if they should be listed or not, or if their current listing was adequate. And so, as many of you are probably aware, this sort of conservation research doesn't have a history of being super well funded. So a lot of the projects we were working on required the use of open source bioinformatic tools to analyze genomic data. And so I became really familiar with using those. And I actually became pretty passionate about using them too, because I think it's just a really amazing concept for open science and also to allow anyone that wants to do research to be able to do research at the same level as any other tool that you would pay for or you wouldn't. So this sort of stoked my interest in these open source tools. And though I was doing a lot of genomics research, I also really enjoyed the computer side, which some people in my genetics lab would say is crazy, but I did. And so when I graduated, I moved down to the UNC Charlotte system in North Carolina, and I'm doing bioinformatics data management here, as well as some training. So that's a picture of me hosting a training at UNC Charlotte. So the lab I work in is run by Dr. Anne Lorraine. She's in the call right now, so if anyone wants to grab her afterwards, please feel free. We have a research lab full of PhD students as well as master's students and a few staff members. Traditionally or historically, Dr. Lorraine has done research on various crop plants, so that's tomato, Arabidopsis thaliana, blueberries, and rice. And, while we were doing a lot of actual genomics research previously, it's shifted more into the software space as we needed to develop tools to actually visualize the genomic data that we were producing. And so now this is sort of the current Lorraine Lab mission or goal or outlook is to create software for scientists like us to visualize and understand their data better. So what I'm going to be talking about today are some of the tools that we create to do this, one of which you may have heard of, which is Integrated Genome Browser, but also some other tools to integrate the browser with other resources like those present in the AgBioData consortium. So there's lots of great genome browsers out there. I'm sure you've used one or two. I've got AgB in the bottom left, but there's also UCSC, JBrowse, and then IGV are some popular ones. And they've all got different pros and cons, right? So we've got some that are completely web-based, and then we've also got some that are only desktop-based, and there's also a select few that are sort of a middleman. So they've all got different features that might make one more popular than the other for people doing different genetics research. But then the question becomes, if everyone's making all these different tools to do the same thing, how are we keeping them compatible and not pigeonholing researchers into these little boxes? So to start, I just wanted to do a quick demo of sorts, show you a video of what AgB looks like. It's easy to get it confused with IGV, which has a very similar abbreviation, but this is AgB. We have lots of genomes that we're hosting, which can be accessed in that species drop-down menu in the top right. There's also a lot of model species here, both plants and animals, in that sort of carousel in the middle. And what's nice about AgB is that there's this fluid zooming feature. So instead of where you have to click plus and minus to zoom in and out and do sort of jump zooming, you can use that scroll bar to zoom in and out on gene models or your data. So here I'm just zooming in to one of those gene models, getting it centered in view so then I can load some data on top of that and zoom in even further. Okay, so there's some read alignments loaded into AgB. So this is your BAM file, your sequence read alignment data. I'm just zooming in even further so we can look at the nucleotide level and show, polymorphisms in the reads as well as how they align to that gene model. So it's a really good tool for sussing out things like mutations, alternative splicing. You can compare different reads and different data sets, so you don't have to look at just one sample at a time. You can look at multiple, et cetera, et cetera. Oh, let's see. So we know a little bit about the people that use AgB, and it's sort of a worldwide tool at this point, which is wonderful, especially for a small software research lab to have produced something like that. A majority of our users are here in the US, but we also have people from all over the world using AgB at any given time. And we know that they are using AgB to make things like figures for their papers. So here I've got some examples of researchers using AgB to create figures for their publications, whether they're looking at ChIP-seq data or RNA-seq data or just traditional sequencing. We also know from a collaborator of ours that AgB can be used to do some error checking with their data as well. So a collaborator, Dr. Gloria Moudet, had some data that was looking a bit odd when she was doing her clustering, her statistical analyses, and she used AgB to investigate what might be going on. And she ended up finding that there was sample switching that occurred at the sequencing facility. And she was able to identify that based on a known mutation in the mutant strain versus the wild type strain. And she could use AgB to actually visualize that, which is what I'm showing here. So to support all of this research that people are doing and using AgB for, we host over 70 genomes as well as their annotations in AgB. This is a picture of our genome dashboard, which we host on the website. And you can scroll through here. It's a nicer UI than the species drop-down menu that's actually in AgB. And from here, you can click on genome versions, that yellow button, and load it from here into AgB. And for a long time, we were actually manually adding all of these genomes. So those 70 plus that I was showing you just there were all added individually, hand by hand, or by hand into AgB. And so that took a lot of data wrangling, fetching. Yeah, it's just a tedious process. And we were grabbing those genomes and annotations from actually AgBioData resources like TAIR, Soul Genomics, MaizeGDB, etc. So all over the web. But as I'm sure you're all aware, these genomic data resources and repositories are growing at such a quick rate that it became nearly impossible to keep up by hand. And there was a nice quote from a short report by the UCSC team that said, "Gone are the days when a new genome for an organism made White House press briefings and news headlines. Instead, several dozen are completed every single day and silently submitted to data archives," which yeah, is true. It's almost impossible to keep up. So this got us thinking, how can we keep on top of adding genomes to make this a useful resource for these researchers without also having to make this just an individual software system that is replicating what other resources have? And so the main topic I want to talk about today are the integrations that we've done to connect AgB with other data providers online, genomic data providers, using their API resources. So the first one I wanted to talk about was a student-led project. So this is Jaya Shravani Siriganidi. She was a master's student in our lab as of last year. And she came to us and proposed a project using UCSC's new tool called Genome Archive or GenArch, where they're trying to add towards 1 million genomes in total to this repository. And so she was wondering, how can we integrate AgB with this resource to grab those genomes and use them in AgB? And so what a lot of students end up doing with AgB when they're doing their projects is to create apps for what we call our AgB App Store. It's sort of like the App Store for your phone, but we've created one for AgB. It serves as this middleman for like chunk of code or a project that the students can push to the App Store, and then it's publicly available for anyone to use by downloading it or adding it to the IGB App Manager, and then they can use it in IGB. So what Shravani did was created a app called GenArc Genomes, and this sort of encompassed that GenArc project that UCSC was creating. And what it does is it grabs all the genomes from UCSC GenArc using their API, and it can add them to IGB via this sort of app connection. So this added over 4,000 genome assemblies and thousands of data tracks, which is amazing compared to the 70 that we had been hosting previously with just our own resources. And so all you have to do is install GenArc Genomes to IGB as an app, and then you type in whatever genome name you're searching for, and you click the load button, the blue load button on the right, and it will add it right to IGB. Oops. Additionally, with UCSC, Shravani had also worked on connecting the genome browser resources to IGB itself. And so this was, UCSC hosts a bunch of data tracks as well. So chromosome bands, information on centromeres, different gene and gene prediction data sets, et cetera, et cetera. And a lot of researchers that use UCSC get really familiar and comfortable with using these data tracks. So it was hard for them to switch over between browsers when the data they needed was stuck on one genome browser. So what Shravani did was integrated UCSC with IGB so that we could pull those data tracks into IGB, and then anyone using IGB could load the same data between browsers. She also made IGB not backwards compatible, but whereas we did the integration with UCSC pulling data to us, we also did the opposite, where we can now push data from IGB over to UCSC. And anyone wanting to use UCSC that wants to use IGB generated data can do the same thing. And in that way, she created a sort of interoperability between the two apps. So that sort of leads me into the FAIR data principles and APIs that have brought us here together. We're a small research lab and funding is not promised forever. And so these software projects that we're putting out ideally will persist after us. And so creating connections and integrations that make these apps able to talk to each other, as well as putting all of our code online, source code, freely available on places like Bitbucket. We're trying to strive towards these FAIR data principles, and we feel we really align with AgBioData's mission in that way. So in that regard, there are actually quite a few AgBioData members that we've already worked on integrating with beyond just UCSC. And I was going to briefly talk about those here as well. And the first of which I wanted to mention was Nick Privart's group at BAR, who worked on the EFP-Seq browser, which is what we've integrated with IGB. This is a browser specific to Arabidopsis thaliana, and it's specific to RNA-Seq gene expression data sets. So you can compare gene of interests in different expression data sets and compare how gene expression levels differ between them. So here I'm just showing a really quick example of what that might look like. The red arrows are pointing out a spot of exon skipping in this specific gene. On the left is a untreated leaf sample data set, and on the right is a heat-treated leaf sample data set. So this is sort of how you would go about comparing data in this browser. And the way we've made it compatible with IGB is we've added this button called Alignments in IGB, where a user just has to click on that, and it will export the data from EFP-Seq browser into Integrated Genome Browser. And then instead of looking at just coverage data, you can actually look at all of the reads that comprise that coverage data as well, and zoom in and look for things like mutations. So that was the first integration we had done there. Second one I wanted to talk about was Galaxy, which I don't think is an AgBioData member quite yet. It might be, I'd have to double check, but it's a really great resource that I wanted to highlight regardless. It's also an open source resource. This one's web-based, and it's mainly used for, like it says here, data-intensive biomedical research. So I found it to be a really wonderful resource for people, especially students getting started with open source bioinformatic tools, because this provides a GUI for those tools instead of needing to work on a command line. So I think it reduces the threshold of getting started into research a little bit, which is really great. So there's two main components to Galaxy. We've got those tools that I was talking about on the left-hand side. We've got all sorts of tools from genomics analysis to, any sort of data wrangling you want to do and file conversions and things like that. You can do it here. Any open source bioinformatic tool you might have used is probably in Galaxy. And when you run those tools, you're usually running them on some data in what they call a history. History just meaning like the list of all the data that you've added or uploaded to Galaxy. So usually you'll upload some here, your input files, maybe that's your FASTA and your gene annotations in GFF format. And then you can run some tools, and whatever you run will put the output in this same history. So it creates a nice sort of data set for you to look at and also to share with other people. And this is where we've sort of integrated AgB to make visualization even easier. So you can run tools in Galaxy, but then how do you visualize data? Well, what we've done is added a button here. So if you click on a file in your history, it'll expand it and show you some information. There's a little graph icon, which stands for visualize or visualizations. And then if you click on that, it'll bring you to a new tab, the visualize tab. And we've got a nice display in AgB link next to display with IGV. And then if someone clicks on view, it'll export that data into Integrated Genome Browser, given that you have the genome you need open, or it'll warn you. So it's pretty intuitive once you work through that. So yeah, it's a very wonderful tool. And they have a really responsive team. They also have a lot of training materials on there. So it's not just how to get started with Galaxy, it's how to get started with specific analyses. So if you're new to single cell sequencing and you don't know how to get started analyzing a data set, they have probably got a training or three for you. So I would highly recommend. Okay, so another integration I want to talk about was Cyverse. This is another sort of popular cyber infrastructure online geared towards research where you can, upload, share, and run, some data analyses on your data sets. Another student of ours, Karthik Raveendran, he developed something called BioVis Connect to link Cyverse with AgB. So instead of the apps like I had mentioned before, now we've developed a sort of web application to link these two resources. So same kind of concept in terms of integrations, but slightly different methodology. And what Karthik made was what you're seeing here. It's this, a website called BioVis Connect. And it sort of looks like Galaxy in the way that you have this history of data. And again, you've got a view an AgB button. And so if you just click on that, and it will export it into AgB right away. So whatever data you upload into Cyverse, now you can view that in AgB at the click of a button. And again, this just saves researchers, like a barrier to entry for visualizing their data. We're trying to make everything more accessible by making it easier and more intuitive to look at what you're actually producing. So one that we're currently working on, or we're in talks with working on, is an integration with Ensemble, which is a rather large database that we would love to integrate with and be able to pull data from. So we're currently in talks with the Ensemble team to do this. They're currently working on a release, like a new beta release. And so once they've got that in the works and their API becomes finalized, then we will be able to actually integrate with them. And now I had shown this figure earlier, and I'd focused more on who was using AgB. But now if we look at the right side of this image, we can focus a little bit on how they're getting there. And so a lot of the views we get on our website is from Galaxy users. It's the most ways that we get views on our website. And there's also a welcome bar user section at the bottom here. So a lot of these are integration-based views. And so we know that a lot of people are using these integrations, and we know that they're working. And so we really want to keep pursuing them because we think it is actually making it a lot easier for people to view their data this way. And so a future direction we wanted to pursue was, finding more databases and resources we could integrate with. And once I had heard about this consortium at ASPB, like I mentioned, I thought it was a really great opportunity to come talk to everyone here and see if there was any interest or possibility of integrating further with other resources and, uh, really trying to make all of this data as accessible as possible to researchers. So if any of that sounded interesting or possibly feasible, and you'd like to get in touch, like my email's right here, pkulzer@charlotte.edu. I also host a weekly help session every Tuesday. So it's sort of like a drop-in hour if people have questions about getting started with IGB, or have specific questions after webinars like this one. So if you're interested, there's some details at that QR code or at the link I've listed there. And I believe all of these slides are also available online at agbiodata.org if you want to view these afterwards. So I just want to thank everyone that we worked with during these integration processes. There were a lot of people from all of these projects that we worked with to get the integrations just right. So we really appreciate everyone that was behind these efforts. And we are happy to say this work was supported by the NIH. And so we've got resources here. There's a lot of them. A lot of these are papers or previous webinars we've hosted where we demo some of these integrations. And we've also, again, got that source code available online. So if anyone wants to view how we've created some of these integrations, look at the actual code itself, you're more than welcome to. And I'd happily encourage it. But yeah, I think that's it for me. So I would happily take any questions if there are any. And thank you for listening. I really appreciate it.